Well, I think we'll get started, and I want to thank you all for coming. And you know, we shouldn't have a sunny day when we're all inside, but and I know there's a lot going on tonight, but thank you for your support of this campus and coming here. And we'll be doing the raffle drawing later. But tonight, Dr. Matthew Paulus is going to give us a very, very interesting talk. And we're lucky, we have fabulous faculty here. I just have to say, we're just outstanding faculty. And a lot of the way we get them is we lure them by our wonderful community. And here you all are, and many more, because of the quality of life. A great place to have a family, right, Matthew? So we're very fortunate to have Dr. Paulus. He has a PhD in molecular biology. He's got several postdoctorates. He's done research on stem cell regenerative medicine. So it's just fabulous to have him here and to do this talk. We also have a lot of support from our advisory council. We have Judith McQuarrie, Shanna Criscola, Chelsea Goucher, and who? Did I miss anybody? So our advisory council is just that. They give us input, advice on some ways in which what we offer, events we have, and provide support from their organizations to us. We also have Jessie Post back here. I took my glasses off. She came down from Juneau. She's the alumni relations manager and is going to help us later on, talk to us a little bit about the foundation and help us with the raffle. And Wendy Horn back there has helped with a lot of the organization. And of course, our tech person, John Long, who's hopefully going to keep this all running. And I hope I haven't left anything out. Oh, our library staff back there, thank you so much. So tonight's talk with Dr. Paulus is Superhero Science, Biology, and Beyond.
Okay, so thanks for coming. First off, it's going to be a fun talk, so I'm glad y'all showed up here, learn some science, talk about superheroes. So if you had a decent childhood like me, you probably had a box of these or several boxes laying around your room somewhere. Or you might remember seeing these guys on the TV on Saturday mornings. And when we were kids, we always used to pretend to be, as smart as Batman or strong as the Hulk or as fast as the Flash or just as crazy as Wolverine, especially if you had some popsicle sticks or something. And a lot of these classic heroes are seeing new life these days, being sort of the stars of these new huge budget movies like the Avengers, which is great. We get to sort of spread this love of superheroes to a newer generation. Everybody gets an updated look. And some superheroes, I guess, are still waiting for their chance. But so superheroes. Why do we have this sort of deep-lying obsession with superheroes? It seems to transcend generations. Our grandparents had superheroes, and we're still telling the same stories over and over to our kids through movies and things. You can actually trace the origin of superhero stories all the way back to probably even before recorded history, but through oral traditions when we talked about legends. We've got Hercules. He's fighting a Hydra up here. We've got Theseus and the Minotaur down here, Perseus with the Medusa, the Gorgon head up there. And our fascination sort of stems from this triumph of good over evil. We like to see the best of ourselves and our superheroes, and we like to feel like in some ways we're able to relate to them and have power over our world. So science has some other theories about why superheroes appeal to us, not only on a personal level, but also on a level of societies and species. So evolutionary biologists have found that biological altruism, which is to say, individuals basically favoring the survival of their species over themselves, sort of self-sacrifice. This is a trait that confers a lot of survival advantages and promotes the evolution or survival of species in the face of genetic challenges. So ants, for example, there's carpenter ants who have worker ants that are sterile. They can't reproduce. However, their job is to basically take care of the queen who is reproductive. And so by basically sacrificing or picking up a job that causes them to no longer be reproductive, they're able to promote the survival of their species by taking care of the queen and make sure she has lots of babies. Other theories include that altruistic individuals apparently appear to be more attractive as potential mates. And this is true in humans. A study found that having good looks actually enhances this as well. So if you're good looking, maybe try to be altruistic. If you're altruistic, well, it's not the end of the world, so there's still hope for you. But whatever the reason is as to why we're fascinated with superheroes, the truth remains that these are really these role models that we're able to relate to. They give us good, positive values that we can sort of try to emulate in our own lives in an effort to grow up with the qualities we see in our favorite superheroes. Plus, superheroes are just fun to talk about, and since it's getting close to Halloween time, it might be fun to dress yourself, your kids, or your pets up like superheroes as well. So feel free to take some ideas from some of the superheroes you see in here. All right, so welcome to the talk. Going to be talking about some superheroes here, superhero science, biology, and beyond. I'm going to discuss a little bit about not only superheroes as they exist, but superheroes, why they exist, what biology has taught us about mutations that might cause superhero abilities, and why learning these things might be beneficial to us. All right, so when I grew up and looked around, one of the questions I asked was, where are the superheroes? Like I said, we grew up watching these on TV, reading lots of comic books. And one thing you take away from all these is that there's a ton of different types of superheroes out there. Basically, there's a superhero for every type of person's interest. And that's probably on purpose. I mean, the designers of superhero literature and products, they're tailoring this experience for basically so it appeals to the general public, so kids can relate to their favorite superhero. Anyway, as I grew up, I became a little disappointed that there wasn't this vast plethora of genetic diversity that I assumed to exist in the superhero world. But just because it's harder to find this in a grown-up adult world doesn't mean it doesn't exist. And we see lots of examples of humans doing super things all the time. And these are things or abilities that we consider to be superhuman in the fact that the average Joe can't really accomplish them. This is Usain Bolt. He's the fastest man on Earth. You can see he has a vast, a large collection of gold medals, lots of Olympics, lots of world championships, lots of world records. He has the world record for the fastest 100 meters, it's 9.58 seconds, and 200 meters in under 20 seconds there. He also holds a record for the top speed ever held by a human running. That was 27.8 miles per hour over, I think it was a 20-meter stretch. Incredible athlete. And just to point out, this is one example of a superhuman, and he also happens to wear some sort of colorful spandex uniform as well. So maybe he's not that far off from something like the Flash. His record falls a little bit shy. Flash's reported top speed is somewhere around 2,500 miles per hour. So a little bit faster than 28, but yeah, still heading in the right direction. There's also individuals like this guy here. This is Herbert Nitsch. He's often referred to as the deepest man on Earth. He holds multiple records, all for skin diving or free diving. So this is diving basically without the aid of diving gear. He holds his breath. Sometimes he grabs a rock and he jumps off the side of a boat. But on June of 2012, he dove to 253 meters, which is about 2/3 the height of the Empire State Building. And then keep in mind, he's got to ascend up from that depth as well. He actually did sustain some injuries from this dive, but didn't stop him from doing what he does. There's actually a separate individual. He's a skin diver that holds the longest breath record. Longest breath ever held by a human was 22 minutes and 22 seconds long by Tom Siedas, which is insane. You could almost hold your breath through this entire talk. These records are impressive. The actual time that Aquaman can hold his breath is unknown. I've looked into it. However, my guess is it's probably longer than about 20 minutes, so he's probably got these guys beat anyway. As far as strength goes, there's lots of feats of strength we see in modern day humans. Here's Paul Anderson. He on the left here, he holds the world record for the biggest weight ever moved by a human being, and it was 6,270 pounds. I don't know if that's actually the lift in that picture there, because he doesn't even look like he's straining, but that is a cool picture that I found of him. Eddie Hall over here just recently broke the world record for world's largest deadlift. He's a weightlifter, bodybuilder from England. And deadlifting is basically lifting this weight off the ground. So it's a little bit harder to do than lifting weights off stools like Paul Anderson was doing. But his deadlift was still impressive, over 1,000 pounds. If you look close, you can see blood coming out of his mouth and nose. Reportedly during this lift, the pressure in his head was so intense that he ruptured blood vessels and collapsed soon afterwards, but still pretty impressive. However, that doesn't pale in comparison to Superman. To find an actual number I could put on Superman's strength, I had to refer to an expert. So this is, of course, going on the internet, hosted by someone named Nightwing123, who actually applied some mathematical calculations and calculated Superman's overall benching max to be 1 dectillion, 50 nonillion, 14 octillion pounds, or equivalent to about an estimated weight of the solar system. And he based this on some feats of Superman in his comics, benching the Earth for 5 days, apparently about 250,000 reps. So still a little bit more than we see in the human world. Humans have special abilities that extend beyond physical prowess, of course. We have individuals that think in different ways. So their brain allows them to operate and attack problems in different ways than us. And these individuals give us a lot in the way of technology, medical technology, engineering, chemistry, medicine. We see names like Tesla and Einstein and Curie and Hawking show up here as well. And these should be thought of as superheroes too. They possess abilities greater than the average person. Even though they're a little bit trickier to quantify, we can definitely say that they are tend to be a little bit more intelligent than the average Joe. And of course, there's a parallel for this in the comics as well. We see a plethora of superheroes that are also scientists, engineers, chemists, inventors of some sort. And they are often ones that possess these superpowers. So if we see evidence of these powers around us in the world, even if they are sort of these muted down versions of what we see in the comic books, it still raises the question of where do these powers come from? So why do certain individuals have abilities that are greater than the average person's? And the simple answer, of course, coming from a molecular biologist, is DNA. So DNA, of course, is this molecule that lives inside of our cells. It encodes all the information that you need to build all the proteins in your body. It's actually a fairly simple molecule in the fact that it's not made up of that many different types of chemicals in there. When we say DNA stores information, what we really mean is the sequence of these 4 chemicals, thymine, cytosine, adenine, and guanine here in different colors, the sequence that those things exist in our DNA basically determines the type of protein we can build from that piece of DNA. So the path to special abilities is not really any different than the path we would take to get to any other ability that humans or anything else that's alive and has DNA would possess. Starting with the information encoded in DNA in that sequence of base pairs, we're going to use that information to build proteins. Those proteins then go do something in the body, and doing something in the body is a good way to, another way of saying that you have some sort of ability. So an example of this would be the LCT gene. So there's a gene in your DNA, a section called the LCT gene. The information in that gene is used to build a protein called lactase. Lactase protein does something in the body. Its function is to break down milk sugar, which is called lactose. And so as long as you've got a working lactase gene, you'll have lactase protein. You'll be breaking down lactose, and you'll have the ability of being able to process and metabolize lactose sugar. All right, so obviously, there must be some differences between the average human and these humans that have some enhanced ability, and that difference probably exists and is encoded in their DNA. When we look at different humans, we actually find incredible similarity between all humans. So if you were to look around the room at the human next to you or the human on the other side of the world, your DNA sequences, so the sequence of those base pairs in your DNA, it's actually about 99.9% the same. We only differ in that 0.1%. So that 0.1% of difference in our DNA sequences is able to encode all the diversity that we see between humans on the planet. So that 0.1% diversity basically must have genes that would differentiate the average couch potato and Usain Bolt. So the reason that we come up with a little bit of difference between human to human has to do with changes in our gene sequence. So we all have probably the same genes in our DNA, but they're encoded a little bit differently. Genes actually can come in multiple flavors, and by flavors I mean they just have a slightly different sequence of base pairs. So on the left you see this gene, it has a G. On the right, in that position, we now have an A over here. So this is what's called a mutation. Anytime you change the sequence of DNA from one thing to another, you call that a mutation. These are actually extremely common in humans, which is why we see all this diversity on the planet. Mutations can involve just one base pair like this. They can also involve multiple base pairs. Also deletions, insertions of base pairs as well. Anytime you switch around the sequence of DNA in any way, it's considered a mutation. When you switch a single base like this has a special name. This is called a single nucleotide polymorphism. It's only called a single nucleotide polymorphism, or SNP, if this variant occurs in more than 1% of a population. And SNPs occur about every 300 nucleotides, which doesn't sound that frequent, but if you consider that the human genome has about 3 billion base pairs in it, this comes up to be about 10 million SNPs in the human genome. And again, these are just the ones that account for or show up in more than 1% of a population. The true value of all the mutations is probably difficult to calculate. But anyway, anytime we end up with a change in DNA sequence, that of course is going to change the protein that's encoded for by that sequence. That protein that's now different, has a different structure to it, might have a different function, and that could alter the ability of that protein. And in some cases, even single nucleotide mutations like this SNP right here are enough to cause a change in function to create something that we might consider a super ability. Yeah, so since we're talking about mutations, one of my favorite superheroes. Here's an individual. His name is Eero Mantaranta. He's a Finnish cross-country skier who lived in Finland. And he was known at the time by several different nicknames, but he was an extremely well-known cross-country skier. Won numerous gold medals, 3 different Olympics he medaled in, and held lots of world records in his chosen sport of cross-country skiing. He also held another record. He was actually the first Finn to test positive for doping, which wasn't really as big of an issue at the time as it is now. It turns out that doping with hormones like Eero was doing at the time wasn't illegal back then. And at least he was in good company. But as it turns out, a study that was done about 30 years later, so in the 90s, on Mantaranta's family, found out that even though he was doping, he probably actually didn't need to be doping to start with. So it turns out him and his family have a mutation that results in them producing more blood cells than normal. So hematocrit is a measure of how much blood cells you have in your blood. So if you were to pull out a vial of blood from your veins, you'd have cells in there. You'd also have a percentage of that would be liquids, so things that are dissolved like proteins and your plasma. In a normal human, a human male, about 40% to 50% of his blood is made up of red blood cells. In Mantaranta, it was somewhere around 66%, and this resulted in an increase of about 50% in the ability of his blood to carry oxygen around the body. So he can move more oxygen around the body. That's what red blood cells do. And more oxygen you have, obviously, the less tired your muscles get. So this is similar to what Lance Armstrong was doing by doping with EPO, creating more red blood cells to give yourself a competitive edge and enhance your endurance. So in this case, this was a genetic mutation encoded in the Mantaranta family's DNA. It was in a gene called EPO-R. The EPO-R gene was mutated, so the sequence was changed from that of a normal sequence to create a mutant erythropoietin receptor protein, or EPO-R protein. The function of this protein was to stimulate the cell to make more and more red blood cells. And of course, his ability that he got was super endurance because his muscles wouldn't get fatigued. The way this works is that, well, actually, this is considered a disorder. So this is what's called PFCP, or primary familial and congenital polycythemia, which is a mouthful. But this is a genetic disorder because it is a disorder, meaning it's not the usual order or functioning of the body, and it's passed on from family members from generation to generation. The EPO-R protein that was mutated is this little black protein up here. Normally, the way this works in healthy individuals is that when you start working out, you're consuming oxygen, your body releases a bunch of a factor called EPO. EPO binds to this EPO-R receptor protein, and that stimulates this cascade of events that accumulates in the production of more red blood cells in your body. So it's a response that has evolved to allow you to adapt to low oxygen. However, in Mantaranta, since his EPO-R protein was mutated, it basically was on all the time. Whether there was EPO there or not, he was sending signals to his cells to make more and more red blood cells. So he had a higher hematocrit in his blood. And of course, that paid off for him. It's a great thing to have if you are an endurance athlete like a cross-country skier. This is actually fairly rare. So PFCP, I think, has only been found in about 116 individuals ever. So I think they came from about 24 different families. So it's actually a really rare disorder. All right, here's another super looking individual, and this is not photoshopped. This is Liam Hoekstra, and Liam Hoekstra, I'm not sure how old he is in this picture, but reportedly at the age of 5, he did his first Iron Cross in gymnastics, which is insanely hard to do. He was at the age of 3, had about 40% of his body with skeletal muscle. And you can see he's a fairly cut individual here. So Liam actually won what we'd consider the genetic lottery. He has what's an ability of super strength because he has a mutation in a gene called myostatin. So technically what he has is a myostatin disorder or myostatin-related muscle hypertrophy. But what this means is that his myostatin doesn't work. So he's got a mutation that causes non-functional myostatin to be made, which is the protein that normally limits the growth of your muscles. So different individuals, of course, have different amounts of myostatin being made, which is why you might not be able to pack on as much muscle mass at the gym as your friend does. But Liam and other individuals that have a disabled myostatin gene basically never get the stop signal for their muscles to stop growing. And so it ends up increasing muscle growth by about twofold in individuals who have this. Decreased body fat and, of course, increased muscle correlates with enhanced strength. It also doesn't appear to have any known medical negatives to this as well. Myostatin is one of these factors which signal outside the cell up here. So this is the inside the cell, this is outside. So normal myostatin floats around, it binds to proteins, and again, it causes the activation of genes that would turn muscle growth off. Liam doesn't have myostatin, so he never activates those genes. His muscles just continue to grow as he works out. And yeah, that's pretty impressive. Another cool thing I learned about this is that this myostatin mutation is actually, for a while now, has been selectively bred into cattle as well. So without doing any genetic manipulation, basically cattle have been bred together who had this mutation. They pass this mutation onto their offspring, create more cattle that have this mutation. So the last word's the beef. Oh, there it is. So this is a myostatin mutated cattle. They're actually fairly common, and you see it has the phenotype that we associate with Liam and his myostatin disorder, muscle hypertrophy, tons of muscle, which of course is good if you are a rancher, because if you're selling meat by the pound, you've got a lot of muscle right there to make some steaks. All right, since we're speaking about muscle as well, scientists have also looked at qualities of muscles. So besides just muscle being there or not, there's different types of muscle. And you can see this in these two different athletes here. These are both world-class athletes. We have a marathoner on the left, a distance runner, and a sprinter here, short distance on the right. Scientists have looked at basically the muscle makeup and the genetics behind these two types of individuals in these professions. And what they found was that there's a variant of a gene called actinin 3. Actinin 3 codes for a protein called alpha-actinin. And there's two different versions of this. You can have the R version or the X version. And when scientists tried to look and see which version was more common in each of these athletes, what they found was that the R version was much more common in the sprinters and other power athletes as well, including soccer players. The X version was more common in endurance athletes and people who weren't athletes as well. As it turns out, they have different functions. So the R version of actinin or alpha-actinin codes for what's called fast twitch muscle fiber, which has a faster speed of contraction and usually results in faster acceleration. The X version of this is more about slow twitch muscle, which works great under oxygenic conditions. So if you've got oxygen around, In an endurance setting, slow twitch is the way to go. If you're trying to sprint very quickly or lift something really heavily, you're going to be working under different conditions. Fast twitch muscle is the way to go. So yeah, we found this are sort of already naturally occurring mutations in these different types of athletes as well. There are a bunch of other fun mutations that have been shown are found in humans too. So I threw a couple slides up here to mention these. Pain insensitivity is a great one. This is a familial condition, so it's passed on from parents to offspring. And it's exactly what it sounds like. You don't feel pain. So there's actually a genetic mutation in one or both of these genes listed up here, SCN9A or FAAH. And these mutations result in your axons, which are your nerve cells, not being able to fire or communicate. And so they completely lack a receptor called a sodium channel receptor that's necessary for the sensation of pain. So these individuals don't feel pain, which can be a problem at times because they don't respond to pain like you normally would. So they can end up with a lot of burns and injuries as a child that normally we'd avoid because things hurt. Flexibility also exists in medical conditions. This is what's called Marfan syndrome. It's a mutation in a gene called fibrillin. And fibrillin encodes a component of connective tissue, which underlies your skin. This mutant version is just shaped a little bit differently and allows you some more flexibility in your joints. It can also cause you some health problems because you can have very poor posture because you're lacking connective tissue that sort of reinforces your joints and allows you to stand upright. There's a good test for Marfan syndrome if you think you might have it. Besides looking sort of stretched out and emaciated, this little test here, you can try to do it yourself. If you try to wrap your hand around one wrist, and if you're able to, I think it's if you're able to put your thumb over top of your pinky, then you probably might want to get checked out for Marfan syndrome. And of course, there's other types of mutations besides physical mutations. Mental superpowers have also been found in experimental settings as well. A number of different genes have been found to have variants or versions that code for proteins that allow you to have enhanced learning, high cognitive abilities, enhanced memory, faster learning, or enhanced problem solving as well. So most of these are actually based on mouse experiments. The exception is the first one up there that's actually been found as a naturally occurring variant in humans. And on this slide, I just grouped what I called survival superpowers. These are mutations that don't give you a physical advantage in performing in sports or necessarily a mental advantage, but they do confer a survival advantage, which is also useful if you want to stay alive. These include genes that give you resistance to, or mutations that give you resistance to things like HIV, cancer, Alzheimer's, radiation, Ebola, arsenic, even aging. They all show up here, and these are all mutations that have been identified as existing in humans. So it's within that 0.1% difference between all the individuals on Earth. All right, so it's easy enough to identify these mutations. If we look around and sequence enough people's genomes on the planet, we can probably come up with a big list, even beyond what I've presented here, of mutations that might be able to give you powers. But what do we do with this information? Is there any way that me, having been born without any of these amazing mutations, might be able to somehow become a superhero? I looked up superhero origin stories online, and I was not disappointed. I was presented with this diagram, and this diagram is great. It says, if you look this up, what it basically says is that every superhero origin story has some aspect of physical training, technology, trauma, and treasury. Batman having elements of all of these. I love this diagram for a couple reasons. Not just because there's Scrooge McDuck and Little Orphan Annie on here, right next to the Hulk. But also because when I hear training, technology, and trauma, this starts to sound a lot to me like my nursing students and what they deal with every day. So if you're a nursing student, you're probably on the right track to becoming a superhero. But what else can you do? So the easy answer is, if these abilities are encoded in our DNA, we just need to change our DNA. So if we change the gene, that changes the protein, it changes the function, and that ultimately changes our overall ability. And we'd be able to enhance our normal abilities if we can adjust our DNA to basically code for some of these mutations that we found already existing. All right, so we already said mutation. Mutation is how you change around DNA sequences. There's a couple ways to do it. You can change around DNA through chemical exposure or radiation exposure. These are great ways to generate mutations. And this is true not only in the comics. These elements have generated all these superheroes up here. This also works in real life. The problem is, if you jump out there and expose yourself to some chemical or radiation that's going to induce mutations, you're more likely to end up with one of these. This is a 30-pound tumor that was removed from a New Jersey man's belly. He thought he just had a beer gut, and he couldn't lose it no matter how much he worked out. So I guess problem solved for him now. But even Asbestos Lady, she was a very short-lived superhero. She basically wrapped herself in a asbestos clothing, and that was her superpower. It made her resistant to heat, but of course asbestos is a carcinogen, so she can't recommend this approach either. So don't go out and expose yourself to radiation. If you damage your DNA, chances are you're going to end up with a tumor. You're not going to end up a superhero, despite what you read in the comic books. So the reason is because these types of mutations are not accurate. They cause breaks and changes in DNA sequence all over the place. So you have to get really lucky to end up basically not having cancer. All right. So is there a way to come up with mutations that are a little bit more accurate and that our cell can handle without turning tumorigenic? And the answer is yes. So we've already talked about some of these mutations existing, but as it turns out, the structure of DNA itself is actually designed to allow for mutations to happen. So this is an important function of DNA. If mutations never happened, we'd never get any changes in DNA sequence. We'd never have any traits that natural selection could act upon. So we wouldn't get evolution. So if our DNA was immutable, if it couldn't mutate, we basically wouldn't be here because evolution wouldn't have happened. So your DNA is already built to sustain a certain amount of mutation. Now, if it gets crazy and it gets too much mutation in there, it will die. Your cell will kill itself, but it can handle quite a bit as long as you're not impeding the survival of the cell here. As it turns out, the most common form of DNA mutation doesn't come from radiation or chemicals. Where it comes from is just your DNA copying itself. So every time a cell divides, which for most human cells is going to be somewhere around 20 hours, your cell goes from being 1 to being 2. It has to copy its DNA so that the new cell has a copy of the DNA, and it makes mistakes when it does that. Like I said, there's 3 billion copies of, or 3 billion nucleotides in the human genome, so that's a lot to copy. A lot of mistakes are made. It's about estimated to be somewhere between 1 in 100 base pairs or 1 in 1,000 base pairs. And if you extrapolate this out, this comes to about 3 million to 30 million per every time a cell replicates. And that's per cell, per replication. So they're occurring all the time. Now, your body has natural enzymes that can repair a lot of these mutations, but they don't repair all of them. And so some of them stick around. And remember, one of these might be enough if it's in the right gene to give you a new function to the protein encoded by that gene, and that could give you a superpower. So this is a little bit more like X-Men, where you already have super abilities encoded in your DNA, and they're basically being derived from a pre-existing mutation. You're not inducing a mutation in any sort of way. And as it turns out, we can actually improve upon this. So besides just sitting around waiting for DNA replication mistakes to happen, we can use a new tool called CRISPR, which stands for something very long-winded. But what it is it's a gene editing tool that's fairly simple to use and extremely accurate. So with CRISPR, what we can do is we can change sequence of DNA very specifically, and we can cut genes out. We can put new genes into places in our DNA. We can fix base pairs. We can change sequences. We can treat this just like a word processing document. We can backtrack, we can delete things, we can put new stuff in there. And the good thing about this is it doesn't go everywhere. We can control where it goes. We can put, edit one gene, leave the rest of the genome alone, edit multiple genes at the same time. And yeah, it's a great tool, and it's being used for a lot of new exciting things. CRISPR is so simple to use that you can actually do it yourself. If you go online, you can find all the equipment, including laboratory equipment that you need to do this for under $2,000, although this is a sale, so I don't know if the sale price is still applicable. There's also a growing database of different constructs that you can order, different CRISPR components you can order that target different human genes. And so we're already using CRISPR technology in human cells in the lab. The next step comes to applying this to a human organism, a living, breathing creature. As it turns out, some scientists are already moving in this direction and experimenting with testing CRISPR technology on humans. This guy is Dr. Josiah Zahner. He's actually the owner of the company that sells the CRISPR gene editing kits over here. It's called the Odin. But he's actually a fairly intelligent individual, a former NASA scientist and current celebrity in the biohacking world. But he has actually generated over here a human myostatin knockout CRISPR system that, if applied correctly, will disable his myostatin gene. And so what he's done is he's injected this construct into his arms multiple times, hoping to disable his myostatin gene, which of course would then allow him to beef up his arms through the same pathway that Liam Hoek's-Druck does. So yeah, it's questionable whether or not this is actually paying off for him. Odds are that this is not a great way to go about it. It tends to be fairly difficult to get CRISPRs to work in an adult because there's so many cells in adult tissue. And you can't really control this, that it's going to stay just in your muscle tissue very well. So if you really want to get CRISPR to work to all of its glory, you got to use less cells. And the best way to do that is to use CRISPR in embryos. So embryos are basically early in the developmental stage of human growth. And so there's much fewer cells. So you don't have to worry about getting that CRISPR system to millions of cells. You just have to worry about it getting it to a few. And so people have been doing CRISPR type experiments in human embryos for a while in different countries. However, this raises a bunch of ethical concerns. So once you introduce the CRISPR system and whatever mutations it encodes into an embryo, and if you allow that embryo to grow into an adult, that CRISPR system spreads to every cell of that embryo's body, so including its germline cells, so the cells that will make sperm and egg. And so these mutations could be passed on to the next generation. And of course, that could influence not only the individual and their children, but on a large enough scale, influence human evolution altogether. So for that reason, there's basically been a large ban on experiments you can do with CRISPR on human embryos. You can see the US up here is restrictive. We can do CRISPR embryo experiments, but we can't implant them. We can't let them grow into fully functional or to term babies, basically. You have to terminate them after a few weeks of growth. Other countries also have restrictions on there. However, in 2018, many of you probably heard this news. This Chinese scientist, He Jiankui, he was the first individual to actually do some CRISPR manipulation on human embryos. He implanted them and they were carried to term. And so they resulted in two females that now have a mutation in the CCR5 gene. And so this is a mutation that should allow them to be resistant to HIV-1 infection. He had altruistic goals in doing this, but of course he didn't follow his country's laws, and the ethical concerns involved with human experimentation are always up for debate. And so he's faced a lot of criticism, and he's might even have some legal charges filed against him as well. So since ethics are on the table here, we've got the ability, the technology to basically move genes from one place to another. We can copy genes with CRISPR into human genome. It's possible, whether it's an embryo or adult tissues. But why stop with human genes? They don't have to be human. All things that are alive have DNA, and they all store up information in the same way. And so that includes everything alive: plants, animals, fungi, bacteria, yeast, anything that you would consider to be alive. And what's also cool is that if you look at sequence similarity between genomes, so DNA sequence between different species, they're actually not all that different. So we're always seeing human to human. This comparison results in about 99.9% identity. Human to chimpanzee is about 96%, same sequence identity. To cat, we're about 90%. About 85% similar to mouse, 80% similar to cow, about 60% to fruit flies, chicken, and banana. It's an interesting combination. But all this suggests that we use the same language to encode things, and much of this DNA is encoding for the same kinds of stuff in these animals, which is to say that it's possible to move genes from one species to another, and they might actually have the intended function in a different organism. Further, besides just the species or the sequence identity being very similar to humans, DNA, the language that DNA uses to encode proteins is also the same. It's pretty much identical for every species of living thing on Earth. So the code is going to be interpreted the same way in our body, whether it's a human gene being read or whether it's a chicken or banana gene being read. So This knowledge has led us to be able to do things like take a gene that codes for a fluorescent protein from jellyfish, put it into a mouse, and we don't create jelly mice, but we create GFP mice. And so these are mice that express that fluorescent jellyfish protein. It's only under UV light, so they don't actually glow in real life. But you can see these guys next to their litter mates who don't express the gene in there. When they're pups, they don't have hair, and so their whole skin's glowing. On the right, they're adults, and they kind of got that creepy vibe with their eyes and nose and ears glowing. But these are pretty cool. And these are what's called transgenic species. So transgenic meaning that we took a gene from one species, popped it into another species. It's coding for protein in the same way any gene would code for protein. It's being expressed in different parts of the animal. And yeah, it's having a function, in this case, causing fluorescence. This is also pretty similar to the backstory for what's called Bat Cow. He's a transgenic cow in the DC universe. So if we have this ability to basically move genes around between species and create transgenic species, why not in the future create some sort of human, transgenic human? Of course, besides the ethical and legal concerns that are limiting right now, it's still a fun mental exercise to think about. And these are some of my favorites as far as species that I would steal genes from. Here's a chitin. This is, you find these out on the rocks down at low tide. Chitin are actually fairly impressive individuals. They produce the strongest substance that can be synthesized by an organic or living being. It's called magnetite. And it looks like this over here on the right. You'd see it's an iron oxide. But what they do with the magnetite is they express it on their tongue, in essence. It's called a radula. But this is what they use to scrape the algae off the rocks. And magnetite's so hard that it actually wears away rock itself. So you can imagine if we could figure out the biological pathway that encodes for this in chitin, and if we could introduce that into humans in a controlled way, maybe we'd be able to generate something like an armor covering, like Colossus here. Another cool thing about magnetite is it forms a powerful permanent magnet. And so you might be able to manipulate metals through this magnetic field if you had magnetite in your body, just like Magneto. Now, vipers, they have the ability encoded in their genes to see infrared. This allows them to hunt at night and they basically see heat, which is a great thing to have. It'd be super cool to have that as a human. If we could figure out all the genes that encode for that, introduce them into humans, we could end up with Superman's creepy X-ray vision. Or like the TSA guy at the airport. Another super individual. And of course, there's the electric eel, which has one of the coolest adaptations in biology. The electric eel generates an electrical current. It's not actually an eel. It's just the name has stuck there. But it can generate up to 860 volts of electrical current in order to immobilize prey. And also, since it causes, as it moves through the water, this electric current causes the muscle contractions in the prey animals. It's also technically manipulating the nervous system of other creatures, which is a pretty sweet ability to have as well. It's kind of complex trait in electric eels. They have a lot of different organs and specialized tissue that goes into creating this current. So it might be a little bit tricky, at least initially, to move this ability to humans. But there's another creature, a bacteria called Geobacter metalloreducens, that actually does a similar thing with much less hardware. So it's a bacteria, it's a single cell, doesn't have any specialized organs, but this thing creates electrical current that it uses to degrade metals. So it shoots out little bits of electricity. And what else is cool about this one is that it fuels itself by basically eating uranium. So that sounds like a pretty sweet a superhero ability. Not only are you powered by uranium, but eating uranium, removing that from the environment would be a great bioremediation service you could be doing while you power your electrical circuits. And of course, one of the coolest mutations or special abilities would be regeneration. So regeneration is actually pretty common in the animal kingdom. It exists in vertebrates and invertebrates. Chop off a sea star's ray, it'll grow a new ray back. Salamanders can lose arms and grow back limbs. Zebrafish up here, I worked on for a while. You can cut their spinal cord, lobotomize them. They will grow back that tissue and be good as new in a couple weeks. Scientists are actively working on figuring out which genes are actually allowing these abilities to take place in these organisms and trying to figure out how to reactivate them or turn them on in humans so that we would have some of these regenerative potential. a lot like Deadpool, or this is how was it? Fall Off Arm Boy. Horrible superhero, but his special power was he'd pop off his arm and throw it at people. So there's a lot of cool mutations out there in the natural world that are already, just sort of ripe for the picking once we get the technology down to deliver these to humans. But we don't even have to be limited by that. There's a field of biology called synthetic biology, which allows us to program genes that don't exist anywhere else in nature. So as long as we know what we're doing, we can design custom genes on with computer programs. We can synthesize the DNA in a lab, and we can put it into a single-celled creature at this moment. This allows us to program bacteria and things to do special jobs for us that they wouldn't be able to do in any other way, like kill cancer cells, detect landmines, detect oil, make drugs, make food, generate electricity, and really anything you could think of. As long as you're a good enough programmer to program the right genes in, you can make a bacterium that does anything you want. So I have Superman up here because in some fan fiction about him, I don't think it's actually canon, but it's claimed that Superman's powers come from synthetic biology. So he's been synthetically engineered to absorb sunlight from Earth and translate that into his special abilities as well. And moreover, since these is fully synthetic, you don't even have to be limited by the existing nucleotides, those 4 nucleotides that occur in our DNA. You can use completely alternative chemicals as your DNA that build completely synthetic proteins so that you can build things, structures, and proteins in your body that do completely new functions. And of course, there's no reason why we have to limit ourselves to just applying one of these powers at a time. We can multiplex CRISPR mutations from humans, from other animals, and some of these synthetic traits as well. They could all be mixed together and delivered all at the same time to create some of the most powerful beings the world has ever seen. Like Super Jock is what I called him, who possesses mutations in myostatin, actinin 3, and the EPO-R. These are those 3 physical mutations that we had. Would basically result in him having super strength, super speed, and super endurance as well. Or the Sinker, maybe somebody who could rival or improve upon some of the skin divers that we mentioned earlier in their records. There's mutations that enhance your deep diving ability, your ability to hold your breath. Those that increase bone density, which not only makes your bones stronger, but makes you less buoyant, so it helps you sink faster. And low oxygen tolerance, so your body can deal with holding your breath for a long period of time. Or my favorite, the Undergrad, mutations that might allow undergrads or whoever has these to have enhanced problem-solving skills. As a bonus, also low anxiety, function with less sleep, have a faster metabolism of alcohol, and low odor production. Definitely a plus if you're working in the lab in close proximity. So yeah, there's no reason why we couldn't do this. We have the technology to. For most cases, as long as we know what genes we want to target and introduce to individuals, the only thing really limiting us is our imagination. CRISPR, these are all mutations that are already found to exist in humans. So all these examples could get a lot crazier if you start throwing in some of the animal and synthetic mutations. You could get as crazy as you wanted to, but these are ones that already exist in humans. We know that they're safe. So they are the logical first line of superhero mutations I would suggest creating. Yeah, I should point out too that the CRISPR system is improving all the time, and they currently are working on a method to make these mutations non-permanent. So you could basically try out these mutations. They affect your RNA instead of your DNA, but they would only be transient and wouldn't be passed to your offspring, which is pretty cool. So maybe in the future you can go to a shop, customize your own superhero CRISPR injection, and have a fun weekend fighting crime. All right, so we're not there yet. We're not making superheroes every day, but we are using some of the stuff that we use, that we learn from studying superhero genes to fight diseases all the time. So basically the other side of superhero genetics are mutations that cause you to have, sort of unsuper abilities. So abilities that are less than the average individual. And these typically manifest as health problems. For example, we're using what we learned from myostatin inhibition, like what we saw in Liam Hoekstra here, and in those super cattle. We can use what we know from that about how myostatin works to develop treatments that inhibit myostatin. And these can be beneficial for increasing muscle mass on basically anyone who needs it. So atrophied muscles, people in comas, or people who can't work out, or are older in age. Also, may be benefited. The research looks like it may be beneficial for treating symptoms of people who have Duchenne's muscular dystrophy. Blood disorders like Mantaranta's, where he has obviously the mutated version of the EPO-R receptor, have been well studied, and it turns out that the EPO receptor plays a huge role in a lot of different disorders. You can, of course, use that mutation that Mantaranta has to try to engineer an active EPO receptor and increase the amount of blood cells produced in anemic individuals who don't have enough blood cells as it is. It's also been found that the EPO receptor activation is important for bioprotective effects on the brain, so it keeps your brain younger as you get older. It also looks like, of course, if you inhibit EPO, you can inhibit blood vessel formation and tumor angiogenesis. So if this is an important step for allowing tumors to metastasize, if you target that receptor, tumors can't form blood vessels, they can't grow large, they can't metastasize throughout your body. And we mentioned the Chinese individuals, the females who were born with CRISPR edits to the CCR5 gene. CCR5 mutation has continued to be studied. And actually, we've had successful human trials using CCR5, not in human embryos, but using a CRISPR-based system to deliver CCR5 mutations to T cells. And what this does is it makes the T cells basically unable to be infected by HIV-1 virus. So the individuals had T cells taken out of their body, edited in the lab with CRISPR to induce this mutation, and then the T cells were thrown back into the human host, and their viral load of HIV basically decreased. All right, so in conclusion, so superheroes, they allow us to aspire to great things. They give us powerful role models that we can emulate in our lives. One thing is, as we continue to try and become more like these models, these superheroes, and speculate about some of the powers we might like to have, it's important to not forget the superhuman powers that we already have. So, evolution has basically selected for the most beneficial adaptations or the most beneficial mutations that existed in our species over time. So not too long ago, it would have been thought of as a superpower to be able to hold a memory in your head, or to look at the world around you and understand what's going on, or to do some math problem, or to ride a bike. These were all things that, in our evolutionary history, our distant ancestors would have viewed these things as being superhuman, and now they're everyday things. So the odds are this trend is going to continue as the environment continues to change and humans continue to evolve to be a better fit, a better adaptation. We're going to see more and more of these mutations become more and more the norm. And eventually, we might actually get to a point where we realize that everyone around us is actually a superhero, and we've got a whole world full of this diverse superhero genetic background. That's my talk. Thank you for coming in here, and I'm going to welcome any questions here. I do want to point out this little link down in the corner here. This is a compiled table of experimental evidence that George Church, who works at Harvard, has compiled online. If you want, you can take a picture of this web address and check it out. But he's got a more complete table of these mutations in humans, as well as links to literature and experiments that support the reported effects of them. So thank you for coming, and feel free to ask any questions. I'll do my best to answer them for you. Yeah.
So one genetic mutation, sometimes a person is a carrier, and if two carriers get together, then there can be it comes out. Yeah. Like sickle cell. Mm-hmm. And I think what I remember is that we've learned that sickle cell carriers are more resistant to malaria.
Yeah, you're right.
But if they get it together, then it's bad.
Right. Yeah, and so what this comes to is a little bit more in depth than I went into here, but everybody's got 2 copies of every gene in their body. And those 2 copies can exist as the same version, or they can be different versions. So you can have a mutation on just one of those copies. So in sickle cell disease, if you have sickle cell disease, you basically have 2 mutated copies of the sickle cell gene. So all of your blood cells will have this sickle shape. It makes it hard to move oxygen around. Those blood cells clot, and it's a bad disorder. But what you can be what's called a sickle cell carrier. You can have the sickle cell trait. And that's sort of the best of both worlds. So you have one normal, healthy version of that gene, The other gene is the mutated sickle cell gene. So you have some cells that are sickle shaped, other cells are completely healthy. So the healthy ones can move around oxygen. The sickle shaped ones, they're not enough of them to basically cause as much health problems as would be if you had full-blown sickle cell trait. And you mentioned resistance to malaria, which is a great one. So if you have that trait, those sickle cells are basically harder for malaria parasite to infect. And so you're more resistant to infection with that parasite. And this is why we see sickle cell trait a lot more common in populations in the tropics where there's a lot of mosquitoes and malaria. But yeah, it's a great example of sort of the benefit side of having one of these disorders. And sickle cell is also one that they're doing first human trials using CRISPR. So in a human, they're trying to fix sickle cell disease in humans. I think it's going to start this year. Yeah, Ken?
Sarcoid. You know anything about sarcoid?
Sarcoid?
Yeah, sarcoid. Sickle cell and sarcoid are kind of eerily related.
Yeah, I don't think I know anything about
I'll
that.
get together with you later. I'll get together with you later.
All right.
I think we might be getting ready to do the raffle, and then you can maybe ask your questions of Matthew afterwards. Or did you have a quick question?
Yeah.
One more.
Has there been any studies done since the Philadelphia chromosome? Has there been anything similar to that causing mutations?
Yeah, so mutations, like I said, they can come in a lot of different flavors. So it can be just one base that changes, or it can be like the Philadelphia chromosome, a whole chunk of chromosome basically breaks off. It's actually it's pretty interesting. Sometimes you end up getting breaks on two different chromosomes, and the little pieces that break off sort of join up and create a hybrid chromosome. And it turns out in leukemia, a certain form of leukemia, this happens, and right where the two chromosome pieces come together, it creates this fusion gene that codes for a functional protein. It's called BCR-ABL. So BCR-ABL basically turns on genes that cause survival, and you can't turn it off. So it causes the tumor cells to basically be proliferative. They survive for longer even when they should die. But yeah, so there are many other mutations that are caused by chromosomal breaks and translocations and inversions and things. Not just these simple mutations.
Isn't a virus a big part of that? Virus moving through RNA to cause the DNA to.
You can viruses can change the DNA sequence. It's actually how gene therapy works as well. So viruses are kind of just packets of DNA or RNA, and they shoot that into your cell, and then that DNA gets incorporated into your genome and expressed. The proteins get expressed that are used to build more viral particles. So what gene therapy does is it tries to introduce a healthy version of a gene with a virus into your cells. The problem with that is that that healthy gene version, it can integrate anywhere into your genome, even right in the middle of a gene that you might need for survival of that cell. So oftentimes treatment with gene therapy can especially with retroviral vectors, results in tumor formation as well. So it's a danger of gene therapy.
Well, thank you very much, Matthew.